these to walk with on the substrate. They use the swim rats to swim, but the most anterior pair of swim rats are modified in the male to transfer sperm. They'll literally make packages of sperm called spermatophores. They'll release it at the base of their fourth walking leg. There's a pore here. They grab those spermatophore packages and transfer it to the female. That's how they mate. We don't have time to do this, but there are a lot of people here at UC Berkeley who study the various genes that are turned on and when they're turned on, and which promoters and which enhancers and various factors that affect this. And you saw some examples of that. For example, in the fruit fly, Dr. Patel showed you, we can get eyes forming where legs were and stuff like that, because you can get differential gene expression. So it's pretty well studied now. In terms of the arthropod diversity, the trilobites look like this. We'll go over that shortly. Horseshoe crab here, so that's the Xiphosaura. The scorpion here. Crustacean. The barnacle, that one's kind of hard to get, but it does have a chitinous exoskeleton. The hexapoda here. Again, here would be a millipede, so that's in the mandibulata, the myriapoda. And since this has 2 pairs per segment, it's the diplopoda, and these are the centipedes, which are the kilopoda. And then just the hexapoda here. And this would be a arachnid in particular, and the crustacean here. And here's just, again, another way to look at this. Let's just look at a few things quickly. I do want to try to show you actual images of the dissections. I think that's probably more useful than me reading off a lot of stuff. But anyway, if you look at the dorsal surface, you can see these black openings here. Those are called spiracles. They're basically valves to the outside. So in this case, gas enters through these spiracles. They enter these small tubes called trachea and subdivide to other tubes called tracheals. And what they do is they have a completely separate respiratory system. It does not interplay with the circulatory system. These tubes diffuse so that they're within about 3 to 4 cell layers from every cell. So gas is just freely diffused from those cells to the interstitial space into those tubes in and out following gradients. So that's how they get gas exchange there. So you'll see those spiracles very clearly on the outside surface. In some cases, people have issues with cockroaches. They try to kill them. They want to do it in a safe manner. So one method is diatomaceous earth. Finally, powdered diatomaceous earth, because it's very crystalline. And what happens is, as those cockroaches crawl through it, it's not toxic to them. It basically abrades their exoskeletons like sandpaper, and they desiccate. Likewise, if you take and use boric acid, the same thing. It's not the acidity, it's the fact that it's crystalline. Okay? And those tend to be pretty safe methods as opposed to using various poisons and that. So we have the respiratory system. We have wings here. On the ventral surface, we can tell males versus females. This is a female here. In terms of the ventral, this last segment here, it's split, and that allows a large egg case to be released. So if you look in the cockroach chambers, you'll see some of these large egg cases. Furthermore, when we've killed these cockroaches, some of the females will release a egg case. You know, when they're being, basically because they had the egg case there, the muscles basically relax, the egg case gets released. So that allows the egg case to be released. The males do not have that. Also, this one's sort of more rounded, and that's pointed. The males have these small structures called styles, but they can break off pretty easily. Not the large ones, because that's shared by both male and female, but some small structures called styles. I tend not to look at those because it's really easy to tell male versus female, because this is a broad morphological difference. In terms of trilobites, it's called trilobites because they have 3 lobes to them. If you look in cross-section, so there's 1 lobe, second lobe, third lobe, and they've been extinct for well over 20 million years, okay? And we definitely see segmentation here, and we see repetition. In terms of the Chelicerata, so we've done the Trilobitomorpha, the one branch off. Now we're doing the Chelicerata. There's going to be 2 groups, Xiphosaura and then also the arachnids. Horseshoe crabs, some illustrations here. This shows you what's called a huddle. There's 1 female here, about 5 or 6 males trying to mate with them to transfer their sperm. They crawl up on the shore, sometimes on the east coast. You can see they do this sort of in mass. That's the way they mate. This is the ventral surface here. These are their walking legs here. They have this protective exoskeleton. They have a compound eye, actually image forming. They have these structures called book gills here. They're like the pages of a book, literally. They just lay on top of one another, and they can fan them and move them out, and that's how they move the fluid to get gas exchange. So those are called book gills because they're external. And we'll see in the arachnids they have what's called book lungs, but they're internal. But again, they look just like that, plates, like pages of a book that overlap to increase the surface area. So I want to point out a couple things here. So I mentioned the compound eye. Now you may say, why would people ever study these? There's been 3 Nobel Prizes awarded to people who study horseshoe crabs. One based upon compound eye. It turns out that the compound eye here is a great model for studying certain human eye diseases. So what they do is they use the horseshoe crab and this compound eye to try to study these various diseases in humans. It serves as a great model because it's hard to do those kind of experiments in humans. Not very ethical. The other thing is these horseshoe crabs, they have an immune system within them composed of amoebocytes. We don't have much time to talk about this, but all these things have to protect themselves from internal infections, basically. So they have amoebocytes, like white blood cells, that crawl around, and they are exquisitely sensitive to gram-negative bacteria. Exquisitely sensitive. And so pharmaceutical companies to this day will test a lot of their pharmaceutical preparations looking for gram-negative bacterial contamination of their preparations. And what they do is people bleed these horseshoe crabs when they come up to the shore. They basically grab them, they'll drain the blood from them to remove the amoebocytes, make a lysate of them, so they can test these pharmaceuticals. So in fact, there are some real drops in these populations now, in part because of bleeding, part because of destruction of habitat. But so there's 1 Nobel Prize because of that work. Another Nobel Prize in terms of the exoskeleton here. How many of you ever had stitches and didn't have to go back and have them removed because they self-dissolve? Yeah, probably the majority of us. Those sutures are typically made from chitin. So there was some work done on this. And the great thing about chitin is it's antimicrobial in nature. So if you make the stitches out of chitinous material, it's antimicrobial, so you minimize infections. And furthermore, your body will eventually dissolve it. Okay? So that was the 3rd Nobel Prize. And I had a GSI. Well, we'll do that with crustaceans. Okay. Here's the arachnids here, the spiders, 4 pairs of legs. There's those modified fangs for injecting venom in terms of toxin in that, and those Chelicera. The pedipalps help them with feeding. Here shows the anatomy of this. Just point out this book lung. Again, it doesn't show this very well. Imagine sheets of paper stacked on top of one another, but you have airspace in there. So they basically pull in air, pass across these sheets or pages of the book for the gas exchange. And they have an open circulatory system. All the arthropods open circulatory system. In terms of the crustaceans here, the pancrustaceans, some examples. Most of them are aquatic, but we do have some terrestrial ones. You may have seen these before. Pill or sow bugs. They ball up for protection because basically they have this exoskeleton here that sometimes they're called roly-polies. Right? They ball up to protect themselves. Crustacean here. Barnacles, these are indeed a crustacean. They have the chitinous exoskeleton. Here shows you it balled up. That's for protection. Here shows you this. So I had a GSI. I checked today. They taught about 10 years ago, and they were from Russia, and they knew that they were allergic to shellfish. Okay, but they were a math major, so they never had taken biology before. So they took this class. It was a challenge for them, and then they suddenly realized that hey, by shellfish they mean crustaceans, and he realized he could eat fish. So he then realized, hey, I can't eat things like shrimp and stuff or crab, but I can certainly eat fish. He just heard the word fish, right? So anyway, so biology can be useful at times. Here's the myriapoda, many legs. You see this? Here's the centipede or kilopoda, and the millipedes here. We used to have live giant millipedes. In the lab, they'd be about this long. Unfortunately, they're hard to get now, so we don't have any. We will have live scorpions and probably centipede there, but we will not let you pick up the scorpion. We may let you handle the tarantula under one condition. If we let you handle it, wash your hands afterwards because they release not hair, but these really small fragments that can be a little itchy. And also, we ask that if we put it in your hand, that you don't freak out and drop it. We end up having about a tarantula or two killed every semester because people drop them from a high height because they just freak out. Okay? So if you want to try it, part of biology is to overcome your fears. So feel free to try that. We'll bring it out for you. When we had these snakes in the lab, that was also a good opportunity for that. Insects, huge amount of diversity, well over 1 million species of just the insects within the hexapoda. Here shows you what's called a honey ant. This ant basically modifies its abdomen and stores the equivalent of honey there, a nutrient-rich source for other ants within the colony. It's a very complex behavior. This thing will weigh 8 to 80 times more than the other ants. So clearly it's defenseless because it weighs so much. So other members of the colony will protect it, but that's basically a huge storage reservoir for those other ants in times of need. So they have really complex behavior, maternal and paternal care. They have mating rituals, things like that. I'll hopefully show you a few movies of this. Again, the chart, you can just go through that. Here's what I meant by the two forms of development. The complete development here, or indirect egg, a larval form, pupa, the adult, amazing dedifferentiation. Here's specialized cells and tissues there. They have to dedifferentiate, become undifferentiated, and then redifferentiate to form this adult organism. So a lot of information being studied about how do genes, how do you do the switch from one pattern of development, remove it, and start a whole new pattern. Question? It should be limited strictly for the insects within the hexapoda, okay? So most appropriately, people do use it for other groups, but they really should only be used for the insecta within the hexapoda, these terms, okay? Yes, question? So what's the advantage of HOLO here? Because this larval form can be in a different environment. So like mosquito larvae that I talked about. So you have the larval form competing for one food source that doesn't compete with the adults. So by partitioning your resources within the environment, you can actually increase your population density. Okay? That's same for some of the chordates, like the frogs, okay? Where you have a tadpole form and an adult form that's terrestrial. Okay? And then here just shows you that the juvenile forms look like the adults. Okay? Here's some images here. Egg, larval, pupa, adult. Here shows you the dorsal surface, the spiracles, very obvious. Ventral surface here, this is the female there. There's the small structures, the styles. Here's we removed the wings. Here you can see the spiracles. You can see each of these terga, or in this case, the dorsal plates. Here's the head, thorax, abdomen. These are called anal cerci. They help detect vibrations. Here's the female. Notice that split has this ridge here, looks like the keel of a boat, allows the egg case to be out. The male is here, but these styles are there. They can break off. Here you can see those spiracles there. The way we're going to have you do this dissection is please put the cockroach in your hand. Yes, we have gloves right there. Put the cockroach in your hand, and you want to take the scissors, cut off the wings. Cut off the wings. I'm going to grab the forceps, set them aside on a paper towel. And then what you're going to do is take your scissors and cut along the margin here, along the lateral margin there, and there. So what you've done, side view looks like this. And this is the one dissection that's actually a little tricky to do and takes a little bit of time. So this is a cross section here. Cut there and there with the scissors. So if this is so you basically cut along like this, in the abdomen, side view, and then you have these plates that go across like this. You'll have two pairs of forceps. If you take the forceps, one forceps hold in the middle there, take the other forceps there, and you peel this off, you'll split there. Typically, you allow this plate to be pulled off to the side, and this one, and then this one, and this one. So you'll eventually end up with this interior exposed, and if you've done a good job of it, if you have a dorsal blood vessel, you can actually see a little line like this, where that dorsal blood vessel was as you pull those plates off, okay? So I'll show you the image of that shortly. Okay? This just illustrates the GI tract. I'm going to go to this image because this is a bit better. This is for the cockroach. So let's start with the GI tract. They have a mouth, they have an esophagus, they have a crop that stores the food, analogous to the stomach. They have a structure called the gizzard that fragments the food. There's actually chitinous plates in there that go back and forth to fragment the food. We have the gastric ceca here for all the digestive enzymes. We have the midgut, these structures here called Malpighian tubules. That's where nitrogenous waste is being produced. In this case, they take coelomic fluid, process it, and form uric acid. So it precipitates out and just gets released in their feces. It goes out the hindgut. Okay? They have a ventral nerve cord like this. They have clumpings associated with these appendages called ganglia. Here shows you a male here. The testes is pretty hard to find, but the conglobate gland is very easy to see, and the accessory gland is very easy to see. And in the female, the ovarioles are easy to see, and the accessory gland is easy to see. And these two look alike in the sense that they're white tissue, but here it's very diffuse, and here they're all clumped together in the male. Okay? And I'll show you images of these. Okay? Here shows the tracheal system I talked about, the spherical here. Some of them actually have valves that can close this off, leads to a trachea, subdivides, so it's very close to all the cells for gas exchange. Okay? All right, let's just, here we remove those plates. Here's the dorsal blood vessel like this. Lots of fat. So once you clean up the fat, it's going to look much better, like this. So we remove the fat. There's still some fat tissue here. Yes, on the lab exam, I could potentially have this image and ask you to identify fat. Okay? It's not a trick question. You should identify fat. Okay? Here's the crop. Esophagus is up here. Crop. This funnel-like structure there is the gizzard. It looks a lot like a funnel. Here's those gastric ceca for the digestive enzymes. We're in the midgut where most of the digestion is occurring because here's the foregut. Fragmentation, storage in the foregut. Here's the gastric ceca producing digestive enzymes. So most of the digestion absorption occurs in the midgut. Here's the Malpighian tubules, those yellow things there. This is all the hindgut here. Okay? Now, the lab manual has this drawing where they show this really thin section called the ileum. It does get a little bit thinner right here and then a little bit thicker here. But I'll show you another image that shows it a little bit better, okay? Ventral nerve cord here. These white tubes all over the place. What do you think those white tubes are? Tracheals. To provide gas exchange. They're silvery like this because they're filled with gas. There's a ventral nerve cord, tracheals on each side. Female with the ovarioles. Here's the accessory gland there. Those white structures not clumped together. Easy to see the ovarioles there. Here's a close-up showing you the gizzard. Notice how many of those tracheals are there. Why would you have a large number of tracheals on the gizzard versus the crop? Why would there be more? It's more muscularized, right? It's doing more fragmentation. It's a lot more active. Crop is mostly storage. And here's those gastric ceca and then the midgut. Here shows you a close-up of this. This is the midgut here. Here's the Malpighian tubules. Here's the hindgut. I don't know if you can see this, but see how that's a little bit narrower than this? So they label it the ileum. I think it's not drawn well to scale in the lab manual. Again, female there. Male here. That accessory gland all clumped together. Conglobate there. Ventral nerve cord. Why would you have trachos on either side of the ventral nerve cord? Lots of metabolic activity, right? You're sending all those neuronal impulses in that. Needs a lot of oxygen for ATP production. Again, the male here. Lots of fat still here. Crop there. Ovariole not very well developed here. but you can still see that there's the partitions, and here's the extracellular gland. Very different than what you saw in the male, and also hemoglobin is obvious. For the crustaceans, there's that chart like that. It just shows you the fused head thorax, the septal thorax. Here's the ventral surface, female, male. The swim rat's modified here for sperm transfer. Base of the fourth walking leg, spermatophores. Female releases her eggs at the second. So some of the crayfish will actually store the spermatophore, and then later she will release the eggs and the sperm from the spermatophore. They get fertilized, and then she fertilizes the eggs, and they get attached to the swimmerets. In some cases, the spermatophores get attached right to the swimmerets, and then she releases the eggs, and fertilization occurs then. Dissection, very straightforward on this. You just take scissors, cut up here and here, and then fastest way, take your hand, go in there, like you're eating lobster or crayfish, go in there and grab that shell off, the exoskeleton. Here shows you a side view. I mentioned the mouth, esophagus. Up here's the stomach. There's two. The cardiac closest to the mouth, pyloric here, and we have these hardened structures here. Okay, and you see these here. So this is the side view. This is the top view. Here's the cardiac ossicle here. Here's that pyloric here. These are hardened to allow muscle attachment internal. So the gastric mill here is internal here. Can't really see it, but it's down underneath there. It's internal, and it attaches to generative force. That's the point of attachment, generative force. And they basically go back and forth like this. And any food that's there that passes past those gastric mill gets fragmented. And when the food is small enough, it goes through effectively a membrane, a filter-like device, then enters the pyloric stomach, and then out the intestine for absorption, and then waste out the anus. Okay? Any questions about the GI tract? We, of course, see gills over here for gas exchange. This is female, the ovary will be obvious. Heart's hard to see. I'll show you some images. The male, the vas deferens is usually easy to see. Tachys is a little bit more difficult. Heart's a little challenge. I want to point out these structures called ostia. These are actually openings on the heart in the coelomic cavity. If you do this dissection, the heart will be right in here. This is a female ovary. There's that cardiac ossicle, pyloric, gastric mill there. These are the mandibular muscles there. They attach internally to the exoskeleton, and what they attach to ventrally is the mandibles. So if you lift this up and pull in the mandible, you'll see this muscle move. Conversely, if you pull this massive muscle here and look ventrally, you'll see the mandible move, because that's the connection. Normally it was internal to the exoskeleton. Gills over here. The heart will be here. Green gland's going to be way up here. Let's look at a couple images. Here shows you the close-up here of those ossicles for hardening. Here shows you internal. This is the green gland. This is anterior towards the ceiling. Green gland here for nitrogenous waste production. These are mostly aquatic, so ammonia. There's this structure that's found sometimes called the gastrolith, and that stores calcium. So since these things are aquatic, they can add calcium to their chitinous exoskeleton to make it that much more rigid. And soft-shell crabs are typically crabs that have just recently molted, and they haven't hardened their shell yet completely. But since they molt, they have to have a storage form of the calcium, so when they do that molt, they can quick get this reservoir and mobilize it. Male here with the vas deferens here. Digestive gland, all this yellow stuff here. Heart's right in here. Here we cleaned it up a bit. Here's the heart. They have these ostia, those openings. If you take a bottle of Ringers, and you take that Ringers and squirt it into these ostia, you'll expand the size of the heart, and you'll start getting that heart to contract again. It's sort of like CPR. You do it manually, you press this, forces the blood out, it returns, you press it again. That's what CPR is doing. Here, you increase the volume, and the heart will take a few contractions. I also want to warn you, if you take flash, sometimes the flash will release electricity, you didn't realize this, that little bit of electricity is enough to stimulate the heart to do a couple contractions. I had a couple students email me about when they did the rat dissection that they thought they saw that. It may have been the case, but probably not with the rats, because they have been dead for so long. Okay? And they have a higher rate of metabolism, so they're not going to respond like that. Here just shows you a side view of the gills there. Here shows you close to the heart with the ostia, ovary in the back, green gland, gastrolith, and that's it for tonight. Oops, one more. Here's the female with those fertilized eggs on the swimmer. Enjoy the lab, and next week we'll do reproductive development and hopefully show you a few movies.